All of you, the question is describe Lac Operon model of gene expression, isn't it? So that is the question we have. What is this Lac Operon? So gene expression, what is this? Regulation of expression of gene is absolutely essential for the growth, development, differentiation, and very existence of organism. That means whatever we have, hair color, skin color, our formation of proteins in our body, our genetic makeup, everything is regulated by genes. We know that. So that's what is genetic expression, expression of genes. So we have positive regulation. The gene is said to be positive when its expression is increased by regulatory element, positive regulator. Negative regulation is like due to the presence of negative that means gene is not expressed. Like for example, RH incompatibility. If you have RH negative gene, RH positive will not be expressed, isn't it? That's what is negative regulation. So whatever we are like looking and whatever our body is performing, functioning, everything is actually regulated by genes. Okay. So in order to understand these genes, there is one actually concept model which is uh, studied, which is called as Lac Operon model of gene expression. We will try to understand what is this. Actually, Lac Operon consists of, first of all, I will tell you what is this is. The regulatory gene this is. The Lac Operon concept is basically Lac Operon is nothing but this is the like sequence of genetic makeup what we have studied in a bacillus bacteria. Okay, so lactose operon. So what is this Lac Operon? See, the operon concept is coordinated unit of genetic expression in bacteria. We are studying the genetic expression in bacteria. That's what is Lac Operon. The concept of operon was introduced by Jacob and Monod. So whatever is given under this, we should study everything better. So Lac Operon is being asked. No, I will explain you this also. I'll explain you till here. Tryptophan operon is not required. Till negative regulation is given. No, so till that point we are supposed to understand what is this Lac Operon. Okay, then. So see here. So this study, that means coordinated unit of genetic expression in bacteria. Like we have coordinated unit of genetic expression in human beings also, no? Because of which the normal formation of all these systems like hair formation, like sickle cell hemoglobin is one of the expression of abnormal genetic expression. I will tell you a few examples of abnormal genetic expressions in human beings, what we studied. Sickle cell hemoglobin, thalassemia, diabetes mellitus type 1. These are the few examples where genetic expressions are not proper, because of which a gene is unable to produce a proper protein. So how did we observe this? Before we have studied in human beings, we have studied a series of sequences in bacteria. Okay, the bacteria is E. coli. The concept of operon was actually introduced by these two scientists, Jacob and Monod, okay, based on their observations. Okay, and they have studied the regulation of lactose metabolism. Like suppose in order to study glucose metabolism, we take into consideration insulin hormone. Similarly, in the bacillus E. coli, Escherichia coli bacteria, we have studied lactose metabolism to check if the genes are functioning properly or not. So this is popularly called as Lac Operon. That means in E. coli, we are studying lactose metabolism. Hence, it is called as Lac Operon. So structure of Lac Operon, now we have to study diagrammatically, then you will understand. See here, beta. So in Lac Operon concept, what we have studied in E. coli, you have to all like write all these things, but a model of Lac Operon in E. coli, you have to practice everything. So we have one regulatory gene, we have promoter site, we have operator gene, and these are the structural genes which are controlling the lactose metabolism, lactose functioning. Okay, what we are seeing. Okay, now see here, this A is one picture and B is one picture. Operator gene is further shown in 4 boxes, rest will remain the same. Okay, in A you are finding the structure of Lac Operon. In B, repression of Lac Operon. C, see this is the repression of Lac Operon we are seeing. Okay, so now what is repression? Let us see. So constitutive genes we have, institutional genes, inducible genes we have. First, let us try to study the structure. So this, what is B? Always you just study here better. First thing is structure is given. Second is repression is given. C is de-repression is given. Let me check the meaning once better. That means this is nothing but a synthetic gene. Okay. So that is the meaning of repression in Lac Operon. Okay. So what is this regulatory gene synthesizing? Like in human beings also, these genes will be coding for mRNA, mRNA will be coding for certain proteins. Here also, this regulatory gene is actually like coding for one mRNA. So this repressor tetramers, basically they are in the form of subunits. Once all these subunits are united, you call it as repressor tetramer. So here that means this repressor tetramer. Now after this point, we have to go with this. What is this? De-repression of Lac Operon. Let us see what is this. De-repression. So what you are finding here, again same, regulatory gene is there, promoter site is there. In the place of operator gene, we are finding like cyclic AMP. Generally, we have cyclic AMP as second messenger in case of our hormonal mechanism, which we will be studying today. So here you are finding RNA polymerase, RNAP. Basically, RNA polymerases we find in human cells also. No, for what is the function of RNA polymerases there? For the synthesis of messenger RNA, ribosomal RNA and transverse RNA. But this mechanism now we are doing in bacteria. So in this re-repressor, what we are finding like CAMP and RNA polymerases are working. So with the help of this mRNA and this repressor tetramer, it is coding and inactive repressor, it is actually like helping for this mechanism of lactose metabolism. How is this? See this, whatever three subunits we saw, structural genes, these structural genes are actually coding for polycistronic mRNA. So with the help of these polycistronic mRNA, these enzymes are synthesized. We know these enzymes are nothing but proteins. So mRNA is already synthesized from this regulatory gene. With the help of structural genes, we are finding the synthesis of polycistronic mRNA, which are coding for acetylase, permease, and beta lactosidase. All these enzymes will be helping in the metabolism of lactose, what we are studying in E. coli. Okay, so this is a model of Lac Operon. Little complicated and twisted, but you have to understand this thing only better in order to write whatever theoretically is given there. Okay. So I will just repeat the structure of Lac Operon. That means we are now studying the genetic makeup of E. coli. In the genes of E. coli, you are finding regulatory, promoter, operator genes. The same operator gene is actually further synthesized by this mRNA, that is repressor subunits, which are transformed into tetramer. So this tetramer will help in the formation of these RNA polymerases. So finally, you will see one big mRNA structure is formed with the help of all this structure. So a polycistronic mRNA, which will be synthesizing these enzymes, which will be helping in the lactose metabolism of E. coli. Okay. So this is initial studies. When these studies were done, before we have done studies in human beings, this was first studied in a bacteria E. coli by these fellows in 1965, for which they won the Nobel Prize also. Okay. So just read once with the help of these pictures. Though this is little complicated, you will understand. Okay. This you need not study better. Actually, like this is again different gene which is coding for adenyl cyclase. Okay. So control of operon by catabolite gene activator protein. This is for the role of glucose, how glucose is being catalyzed inside this bacteria. Some other genes which are operating this mechanism. Okay. Initially it was for lactose, now this is for glucose. We have different genetic information which is coding and which is helping for the digestion of this glucose in case of this bacillus. So try to read this with the help of these diagrams, you will understand. Okay. So that is for lac operon, and urea cycle we have already done many times, isn't it? These except for this question remaining are very simple. Renal function test. So what are the renal function tests we have? Creatinine clearance test, urea clearance test, isn't it? Metabolic alkalosis we did, alkaptonuria, homogentisate oxidase is the deficient enzyme, isn't it? We did this also. Thyroid hormones, yesterday also I told you. Same thing what you studied yesterday, you can write here. Post-translational modifications. Actually, translation we will take as an essay because Niharika didn't join today, no? Again, she will ask me to repeat. That's the reason. Translation we will take as an essay when we do paper 2 again day after tomorrow. Okay. So functions of calcium we know it helps in like blood clotting mechanism, muscle contraction, neuromuscular junction transmission, isn't it? Nerve muscle physiology and neuronal junction transmission at synapse also we require calcium. So many functions it does. Tumor markers also we did. Can you name few tumor markers? CEA, carcinoembryonic antigen, which is utilized to check some cancers like breast cancer. There are many markers to diagnose cancer at a very initial stage. Serum urea and creatinine normal values will be given on your cover page. Last page of your book you can check better. Allopurinol, I told you hypoxanthine to xanthine, xanthine to uric acid conversion is prevented by allopurinol. So in order to treat gout, we make use of allopurinol. Okay. Four second messengers. This we will see better. Second messenger. Second messenger is one I know is cyclic AMP, CAMP. What has come here also when we were doing this. Apart from that, just four names he is asking. CAMP is second messenger. Similarly, any four second messenger names are asked. Let us see. I'll share in the group. Okay, beta. I'll take the screenshot and share. CMP, CGMP, inositol triphosphate, diacylglycerol. Any four examples are asked, and I have shared those names in our group. You can check. Cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, and calcium. These are the examples of second messengers. Okay. So we will stop here, beta, because many questions in biochemistry are more or less